Good evening. Welcome to our third week of Biotech 201, the biomolecules of life. We have so much to cover this week. I am so excited about what we are going to talk about, and we have another taste test. So for those of you in the room, you've got bags. For those of you that are watching from home, this is the chance for you to run to your pantry and grab table sugar and honey and any artificial sweetener or stevia that you might have, and we will pull that out in just a little while. So before we get started, I just want to say a special word of thanks to those of you in the room and those of you that are watching from home. You were so generous last week. This group collectively donated more than $1,000 for education outreach, and I am just so grateful. Thank you. Thank you so very, very much. I appreciate that. We do not take that lightly, and our job is to be good stewards of the dollars that you provide us with. So thank you. Okay, so we have a lot of ground to cover tonight. We're going to start. You know how much you love the Jackson Center cookies, and you will get them at break tonight, I promise. What makes these cookies so delicious? A, Jackson Center magic. B, buttery lipids. That should be, yes. C, a blend of mono and disaccharide carbohydrates. And D, all of the above. The answer is, of course, D, all of the above. A little Jackson Center magic, just absolutely spectacular. The Jackson Center, along with Orange Beard and Fantasy Playhouse, are our in-kind sponsors. The Ashburn Foundation, of course, is our presenting sponsor. But yes, these cookies, absolutely spectacular. We've already talked about lipids. This week we're going to talk about carbohydrates and about sugars and starches and polysaccharides. So carbs, these are your sugars. They're polymers. This is a word that we've used last week. That means that they are repeating units of something that are strung together. They are starches and your fibers. We'll dig deeper into that. They are composed primarily of carbons, hydrogens, and oxygens. Those are the three molecules. You've seen these atoms before. We've got our molecule of glucose, sugar up here that we'll dig into in just a second. And the monomers are anywhere from 3 carbons to 7 carbons arranged in a specific order. And again, our goal is not to memorize structures. Our goal is to look for patterns and to do comparisons. So that's the level of science that I want us to focus in on. They serve as fuel, and they serve as structural building materials for our cells. And I want you to hold those two pieces. Fuel, so your sugars, your energy, and then structural building pieces. We'll talk about each of those in just a little bit, and we'll connect it to why I am dressed as if it is the end of the 1700s, the beginning of the 1800s. We have another fantastic wig. Never has my hair been this long. Never will my hair be this long. But anyway, so just a lot of fun. So with that, let's go ahead and let's ground ourselves in a little bit of history. So, sugar. Sugar in Europe was known as early as the 1200s. It was rare. It was expensive. It was considered an exotic spice. It generally came from the Silk Roads, so often from India or from Asia or perhaps the Middle East. And it was used primarily as a medicine or to disguise the taste of really bad medicines. But the only people that had access to sugar were the very, very wealthy. 1493, Columbus's second journey to the New World, he takes with him cuttings of sugarcane. Now, sugarcane has at this point, by the late 1400s, is being grown by the Spanish in the Canary Islands off the coast of Spain in and around the Mediterranean, and Spain controls the sugar market at this point. So Columbus takes with him sugarcane plantings, and on his second voyage to the New World, he plants them on the island of Hispaniola. And over the next 250 years, starting with Spain and then with England and France and the Dutch countries, they all began to plant sugarcane in the Caribbean, in the area called the West Indies. So that's the Bahamas, that's the Caribbean, that's the Greater and Lesser Antilles, in the area of South America around Brazil. This area becomes the hub of sugarcane production, and there are enormous sugarcane plantations that arise all throughout the Caribbean. Sugar becomes more plentiful. It becomes something that is not simply a luxury for the rich. It is something that is available to everyone, and sugar consumption dramatically increases. So this is a paper, a booklet, a treatise on sugar that was published in 1799 and then re-updated in 1800. Apparently there was a lot of new stuff in that 12-month window. Benjamin Moseley, Dr. Benjamin Moseley, the physician that wrote this, spent 18 years in Jamaica working in and around the sugarcane plantations. And most of this book is written talking about how sugarcane is cultivated, where sugarcane comes from, and the medicinal properties of sugar. The medicinal properties of sugar. So yes, many people, I know that, I knew that. So this is sugar consumption in England, starting from the 1600s when it is truly a luxury. The 1650s, it said that Queen Elizabeth I, that by the time she was an older woman, her teeth were blackened from tooth decay from eating sugar. You don't see a lot of smiling in those pictures. That's probably best. But so by the time we hit 1700, the average consumption per year is about 4 pounds per year per English man or woman. By the time we hit 1800, that's gone up to about 24 pounds. And by the time we hit 1850, we're now at about 36 pounds. By the time we hit 1910, we're at 100 pounds of sugar per person per year. Yeah. So sugar consumption drastically increases in this 1700 to 1900 window as sugar goes from being a luxury to an everyday necessity. During this window of time, the 1700s and the 1800s, sugar is incorporated into all sorts of candies and baked goods, especially around the holiday. We still carry that into our holiday traditions. Think gingerbread, think honey glazed or sugar glazed hams. Those are traditional holiday sweets that originated in Europe when sugar began to shift from becoming a luxury to something that you had at least on special occasions. As well, in the 17 and 1800s, tea and coffee became incredibly popular, especially in England, and those are relatively bitter drinks. So how did we make them more palatable? Sugar. And by this point, by the late 1700s, early 1800s, sugar is so common and is no longer a luxury that even the poorest laborer takes sugar in his or her tea or his or her coffee. So it is now just an everyday part of life in England and really across the rest of Europe. Oh, let me take a step back. This in the center image, this is the way that sugar was actually sold. So sugar, we'll talk a little bit about how sugar is processed from sugar cane, but sugar was packed into these cone shapes and wrapped in blue paper. And in fact, the cone shape was the symbol for the grocer for much of the 17 and 1800s in Europe, and that was from the sugar cone. It was usually in this blue paper, and these right here are scissors that specifically clipped off or broke off pieces of sugar. They were called sugar nippers. And so you would buy a cone of sugar. It might be a small cone. It might be a very large cone. Some cones might be very, very pure and white all the way through. Other cones might have white on the outside but have other impurities on the inside. But this was the way that you took your daily sugar, by clipping off pieces of the sugar cone and then putting it in your foods or serving it to your guests. From 1740 until the 1820s, sugar was Britain's #1 import. It was their largest import, sugar, mostly coming from the West Indies. Now, in about 1820, sugar's place at the top of the scale was replaced by cotton. So cotton had been known since about the 1200s. The the nation of India had quite a thriving textile industry producing cotton cloth and cotton fabric. And in the mid-1600s, it was introduced to Europe, especially to England. It was relatively cheap. People fell in love with the patterns, the calico patterns, and everyone started buying cotton. And at that point in time, what they were wearing was primarily linen or wool, which we talked about last week. And this run on cotton suddenly made the linen and the woolen producers in England very nervous. And so they complained, and Parliament passed a series of laws called the Calico Act. And the Calico Act forbade the import of cotton from any country and forbade the sale of cotton. So in an attempt to try to suppress cotton imports and to protect linen and wool, cotton suddenly became incredibly scarce, which meant everybody wanted it all the more, and people found ways to evade the rules, and people like flaunted it. They wore cotton, and they were like, well, I'll pay the fine because I am rich enough that I can actually wear cotton. So what ultimately happened was in the late 1740s, 1750s, the mills, the weavers of England began identifying ways that they could craft cotton at a faster rate than they could craft wool or linen, at a cheaper rate, and in much more vibrant patterns. And this process dovetailed with the rise of the Industrial Revolution in Great Britain in the late 1700s and in the early 1800s. And this was a window when innovation was just incredible. People were figuring out how to do things faster and cheaper, and then how to use the power of water and then the power of steam. So you had machines that were now doing the work that had been done by laborers, by day hands. And literally, within about 40 or 50 years, the economy of Britain completely transformed. And by the 1770s, all of the calico rules were abandoned. They were repealed. And at that point, Britain was now the world's leading producer of cotton fabric and was shipping cotton fabric, finished cotton fabric, around the world. There are some key steps. There are 5 key steps in the production of cotton once you start with cotton that has been collected in the field and de-seeded and cleaned. So the first set of steps are these 3 together: carding, combing, and roving. And essentially what they do is they take the cotton fibers, which run every direction, and they align all of the cotton fibers. So the cotton fibers are parallel and they run in the same direction. So that's what carding does. And these giant machines take all this cotton and they feed it in these giant roller drums that have pins on them, and they pull the fibers in the same direction. And then in combing, they literally comb through and they pull out the short fibers, so you're left with the long fibers. And then there's more aligning that happens in roving. And so these are images from the Industrial Revolution of what it looked like in these textile factories that really developed in northern England. Northern England was a great place for this. Number one, they had access to a lot of coal. So for steam engines, it started with a lot of water and access to a lot of coal to run the steam engines. And it would have a damp climate, which meant that the cotton fibers clung together. There was less breakage and there was less cotton fluff in the air. So here is the carding machines and the combing machines, and it comes off in what looks like yarn, but it's really not yarn. It's almost thin pieces of just fabric of cotton fibers called roving. And if you pulled on the roving would come apart. So there was no strength to it. So you then had to take the rovings and you had to turn them into yarn. You had to spin them. And so these were called spinning mules. And what happened is every single one of these, all the way down, maybe it's easier to see, every single one of these lines is an individual piece of roving that is being, this is on wheels here, and it's pulled out, and the roving is unwound from a bobbin, is twisted and stretched, and the twisting and stretching is what gives the roving, turns the roving into yarn. It gives it its strength. And then this entire piece shuttles back in, it's wound up, and then it pulls back out, and there's the next piece that you're turning this into yarn. And so you had one individual that was I think this is called the minder. They made sure that this continued working, and then you've got two individuals that are working on any places where there are threads that are breaking. If a thread breaks, they have about 30 seconds to find the breakage, retie it, and keep moving on to the next one. So this is just continually moving. And then once you've got your thread, you then are ready to weave it. And we talked about weaving a little bit last week when we talked about silk. The difference being that because of the Industrial Revolution, we now actually have what are called power looms. And so these looms are automatically shuttling the thread back and forth. Weaving, you are joining two threads at right angles to each other, and you are passing a shuttle that's got the thread running long ways in between the threads that are running north and south. And so these power looms would continually raise and lower alternating sets of threads as the shuttle moved back and forth between them. So these pieces, the spinning mule and the power loom and the rovers and carders and combers, achieved 100-fold increase in the amount of thread that could be produced per hour compared to people that were doing it by hand. So the Industrial Revolution in England drove the textile industry. And so by the time we get to 1840, Britain now sits at the top of the cotton manufacturing. And that's a position that they held for almost 100 years as kind of the dominant player in textiles. Okay, the key that ties these two together is that sugar and cotton are made of the exact same molecule, glucose. So that's a commonality between the sugar that we eat and the cotton threads in the fabric that we wear. Chemically, they are the same molecule with one small but really important difference, which we will connect to in just a second. So glucose, the most common monosaccharide. Mono meaning one, saccharide meaning sugar. So you're going to see that our sugars come in units of 1, in units of 2, and in units of hundreds to thousands. So monosaccharide, one sugar. This is the glucose molecule. We are not going to put a ton of energy on all of the positions of these things, but you can see 1, 2, 3, 4, 5, 6 carbons. And in green, and on your handout, you can see that I've numbered them. I've numbered them so that those of you that are really interested in how some of these processes take place, the numbers help you orient where you are as we talk through these pieces. And then we've got a C double bond O, and then we've got OHs and hydrogen groups scattered around the rest of it. Now, in most water-based solutions, and certainly inside our cells, glucose adopts a slightly different structure, which is behind me. And we'll talk about how that structure forms in just a second, but some key pieces. Glucose is the major nutrient for your cells. It is the energy source. It is how your cells get energy. They break down glucose. They extract energy from glucose by breaking it down in a series of enzyme reactions. And the students that are in the room and the students that are watching, you know all of those, the process of cellular respiration and all the enzymes involved in that. We are not going to go through that in this room. That makes all of you very happy, I know. But just suffice it to say that by breaking down these bonds, you can pull energy in the form of what ultimately becomes ATP, which is the chemical currency for how our cells run. And then the carbon skeleton chains that are left over form the backbones of lipids, which we talked about in week 1, and amino acids that we talked about in week 2. So you take in carbs, you take in sugars and starches, your body pulls energy from them and takes the leftovers and turns them into the chemical structure of lipids and of amino acids for your proteins. So as I said, in your cell, in water-based solutions, glucose doesn't have this kind of structure. It has a ring structure. So all I've done is I've turned the molecule on its side, and now you're going to see we're going to begin to fold it up, and then we're going to take the OH on the 5th carbon, and we're going to form a bond with this first carbon. And then just for simplicity, I've just taken off all of the hydrogens. So this is the same molecule, I've just made it easier for us to see. Now that same molecule is right here. It's a little challenging to tell. This is the most complicated molecule we've had so far. Oxygens are red. This oxygen right here is that oxygen right there. And then there's a carbon, and then there's a carbon that sticks out. This carbon that sticks out is that carbon there. And then I've got all of my other carbons in this cycle around them. So carbon 1, 2, 3, 4, 5, and 6, and then there's my oxygen. And then you can see that we've got some of our OHs that are below and some of our OHs that are above. And the ordering of those is important. But again, we're just going to talk about patterns. There is no test on this. You will not be required to draw the structure of glucose in order to get your cookies tonight. I promise. But now let's compare glucose, and I've shown you in its straight chain and its ring form. Let's compare that with some other monosaccharides, some other sugars. So let's start with fructose, which is probably a sugar that you've heard before. Fructose is also a 6-carbon sugar, and you can see I've just highlighted in red the difference. And that's that the double bond is at the top in glucose, and it's 1 carbon down in fructose. So that means when it assumes a ring structure, instead of a 6-member ring, it's a 5-member ring. So it has a slightly different shape. It's still a sugar, it's still a monosaccharide, but it has a slightly different shape, and it has a slightly different flavor profile for us. Fructose is more sweet. Our body senses it as more sweet than it senses a molecule of glucose. All right, let's look at one more, galactose. Galactose is a 6-carbon also, so all 3 of these are 6 carbons, but you can see the difference is that the OH in glucose, in the way I've drawn it, is on the right-hand side, and it's on the left-hand side for galactose, which means it's below the ring, below the plane in glucose, and it's above the plane in galactose. That's a tiny change, but that is a profile difference. Galactose is a different sugar. It has a different sweetness. It actually is much, much, much less sweet to us than glucose. Just that little shift in the position of where those atoms sit. Okay, so we've talked about monosaccharides. That's one sugar. We've talked about glucose, fructose, and galactose. Now let's do some combinations. Let's combine some sugars. So that gives us a disaccharide. That's 2 sugars put together. The easiest example would be 2 glucoses. Right here, let me come on this side. 2 glucoses, and you've seen this before. You probably by now know what it means when I put a blue square around OH and H. That means we're going to pull them out. We're going to form a molecule of water, and this oxygen is going to be left now to bind to the carbon, and so it's going to join these 2 glucose molecules together. So we've talked about different kinds of bonds. This instance is called a 1,4-glycosidic linkage. Not a big deal, don't want you to worry too much about it. It simply means that we have joined the 1 carbon of one glucose and the 4 carbon of the other. So it just tells you something about that bond. This is maltose. This is also called malt sugar or beer sugar. It's used in brewing. Let's compare that to a different disaccharide. Let's look at lactose. Lactose is milk sugar. It is found in milk. It is a glucose joined to a galactose. So the sugar present in milk is slightly sweet, but is lactose, and this is how the galactose and the glucose molecule bind to each other. If you are an infant mammal, you have an enzyme that breaks this linkage and allows you to then use this glucose to get energy, and another enzyme lets you convert galactose to glucose to get energy from it as well. So one of the primary nutrients in mother's milk is lactose, and the body, the nursing infants of most mammals, break that apart and then can use that to get sugar from, to get energy from. Now, for most mammals, once they're weaned, Once they've reached that weaning age, the enzyme that breaks this bond shuts off, and they're no longer able to extract energy from lactose from milk. The majority of humans throughout human history, that enzyme shut off when we were about 3 years old, and we had no need for milk anymore. We were not nursing. In pockets of the population around the world, as humans evolved, the enzyme didn't shut off in 2 and 3 and 4-year-old children. And you can see there's a benefit to that, because that means as teenagers and as adults, you can continue to drink milk. Maybe it's cow or camel or goat or sheep, but you still have the enzyme that's functioning to break this and then get energy from both of these. So people that are lactose tolerant, you can break this apart and you can get energy from. People in this room that are lactose intolerant, you are the traditional. The lactose tolerant people are the mutants. You are the traditional, you're the wild type, which means that if you take anything that's got milk or dairy in it, you're going to have an unhappy tummy and a whole host of other issues, because you can't break this apart and this becomes an irritant. Unless, of course, you take lactate or some of those enzymes, or you buy milk that's already got this enzyme included in it. But that one change, spoiler alert, that's a DNA change that we'll talk about next week, means that as adults, you're able to break lactose into its components and extract energy from them. But most of the human population can't. All right, and then one more disaccharide that you're really familiar with, and this is glucose plus fructose, and this is sucrose. This is table sugar. When we think about sugar, when you and I say sugar, this is the molecule we're traditionally talking about, sucrose. Sucrose is a disaccharide. It's a molecule of glucose bound to a molecule of fructose. So lots of history bound up in this molecule right here. Sources of sucrose, 2 primary ones, sugarcane and sugar beet. Sugar beet really didn't become a source of sugar until the late 1700s, early 1800s. And even today, about 2/3 of the sugar of sucrose, of table sugar, comes from sugar cane, and only about 1/3 or less comes from sugar beets. And then the other sugar that you're probably familiar with, because we hear a lot about it, is from corn. And so this is corn syrup, or high fructose corn syrup, which also comes from corn syrup but has a higher concentration of fructose in it. So the sweeteners in the world today, and yes, there's honey and there's stevia and there are all sorts of other ones, but the primary sweeteners that are commercially extracted and made today come from the 3 things that are on the screen behind me. We are not going to spend a lot of time talking about how we extract those. I've put this slide in your handouts to give you the background if you'd like to dig deeper into it. Essentially, you are crushing the sugar cane and extracting the juice, and then you are boiling and purifying and purifying and purifying the juice, and each time you are purifying it, you are getting purer and purer crystals of sugar. You're leaving things like molasses behind. With sugar beets, you slice the sugar beets and then you you run water through the sugar beet, and that pulls the sugar out, and then you go through a purification process. The process for corn syrup is completely different. We're going to talk about starch in just a second, but starch is a whole string of glucose molecules stored in the kernels of corn, and you heat the corn, and then you extract the starch away from the gluten, and then you use enzymes to break that string of starch apart into individual glucose molecules, and then you take more enzymes to convert the glucose molecules to fructose. So that's all done chemically. So again, we won't dig any deeper into it than that, but if you want to walk through the process, this slide provides you with some of that background. This is worldwide sugar consumption. I showed you England's sugar consumption earlier. This is worldwide sugar consumption starting in about the 1820s and then running all the way up. Here's the pounds per person per year. You can see we peaked in about 2000, the late 1990s, the early 2000s at 150 pounds of sugar per person per year. And we've begun to drop, but we're still well above 100 pounds. And then the image behind me actually shows you the makeup of what those pounds of sugar comes from. The blue line is table sugar. That's primarily cane or beet sugar. That's your sucrose. The green line is high fructose corn syrup. And you can see high fructose corn syrup increased. And then at about 2001, 2000 and maybe the late 1990s, its use began to decrease. And then the gray line is other sweeteners like honey, for example, or maltose. So in our society, we have a love affair with sugar. Sugar is in everything. Have you looked at your list of ingredients? Sugar is in your toothpaste. Let's think about that a second. Why do we brush our teeth? To prevent tooth decay from sugary sweets. I know there are lots of other things as well, but why are we putting sugar in our toothpaste? So it tastes good. Yes, but why must everything taste good? Yes, and how good does good really need to be? You know, a can of soda pop, a can of, has 16 teaspoons of sugar in it. I mean, that's just crazy. It's an enormous amount. We are a sugar-addicted society, and I say that as probably having the largest sweet tooth here. Again, this is a no judgment zone, so if you go back to the cookie table and you take 5 cookies, well, let's not do that, okay. And also, the price of sugar fluctuates wildly from year to year. And so you can see, this is the price of sugar. And you can see times that it's peaked during the Napoleonic Wars, during the Civil War. Huge purchases were made by the Soviet Union and Middle Eastern countries in 1974, and sugar went up to almost like 80 cents a pound. Today, sugar hangs out somewhere, I checked the sugar prices yesterday, somewhere around 15 cents a pound. So the sugar price fluctuates wildly, and that, together with our push to be low sugar and to be diet conscious, has led us to create a whole host of sugar substitutes. And these are the sugar substitutes, some of the more popular ones. The FDA has approved 5 sugar substitutes, low calorie, no calorie sweeteners, and they're on the screen behind me. You would not, just looking at them, see any commonalities between them. The way that our body registers sweet, our taste buds have little protein receptors that sit on the outside of the taste bud, and when something chemically binds with it, it triggers a signal that tells our brain, oh, that's sweet, that's sour, that is bitter. And there's a specific combination of the way that molecules bind with that sweet taste receptor, but just in looking at these molecules, you wouldn't necessarily find anything in common between them. We'll talk a little bit about these. I just want to point out sucralose. Sucralose, this structure is really similar to table sugar. So here's a fructose. This is actually a galactose instead of a glucose. And you can see 1, 2, 3 places where we've taken OHs, oxygen hydrogens, and we've replaced them with chloride. And that replacement with chloride doesn't affect the way we register it as sweet. In fact, it makes it even more sweet to our taste buds, but it doesn't get digested in your body the way a molecule of sucrose would. So the vast majority of it passes straight through your digestive system. And that's the key for most artificial sweeteners. Your body doesn't break them down. Or in the case of aspartame, it breaks them down. Aspartame is 2 amino acids joined together. Remember amino acids? Those are the building blocks of proteins that we talked about last week. These 2 amino acids are joined together in a way that actually triggers a sweet sense. And when your body eats aspartame, you just break it down into the 2 composite amino acids. No calories. So, lots of different types of sweeteners. Stevia is a natural sweetener. It's found in the stevia plant, the leaves of the stevia plant. It has multiple glucoses attached to this central molecule right here. So these are all sugar substitutes. These, saccharin, aspartame, neotame, and sucralose, are artificial. They are man-made. Stevia, the molecule, the sweetness molecule is abbreviated Reb-A, is a natural one. It is not artificially created, but it is a non-caloric or a low-caloric sweetener. Which brings us to how sweet these are. So this is a sweetness level using sugar, table sugar, as 1. So if you see a sweetness level that's less than 1, it is less sweet than table sugar. If you see a sugar sweetener that is greater than 1, it is greater than table sugar. Everybody with me on that? So for example, honey is composed of a mixture of sucrose and fructose. Remember I told you fructose is sweeter than sucrose. So honey is a little bit sweeter than table sugar. Agave nectar, which is 50 to 90% fructose, is sweeter. Maple syrup is less sweet. Lactose, this is the sugar in milk, much, much, much less sweet, barely sweet. Let's look at stevia, 250 times as sweet as table sugar. Aspartame, which is also called the trademark NutraSweet or equal, 180 times. Neotame, which is a version of aspartame that is produced 10,000 times sweeter. 10,000 times sweeter. Your sweetness taste bud is on overdrive. It continually is rapidly sending a signal to your brain, and there's some suggestions that that is a challenging, that confuses the brain, that there is so much sweet overload taking place there. Saccharin, 350 times, and sucralose, Splenda, 660 times sweeter. So, with that, let's taste some sugars and some artificial sugar substitutes and some natural sugar substitutes. Okay, all of the things I'm going to tell you are in your handout, so put your handout below you, and I'll answer your question in just a second. Put your hand out below you. Don't open up your bag. You guys know this routine yet. Let me tell you what's in the bag first. But let me stop and take a question.
How much you know what the dance of the millions was in 1920 for the sugar?
What was the dance of the millions? You know, I really should have looked that one up. I looked all the rest of them up, and I just let the dance of the millions go. So I'm going to put out a call to my moderator, to Michelle, and ask her if she'll look up the dance of the millions and why that caused a run on sugar. So I'll let you know what that is. Yes, and then we'll come right here and then right over here.
That neotame you mentioned, that's not something I'm familiar with. Is that available commercially?
So neotame is right here. So it's an aspartame molecule with an additional, with this component next to it. It's marketed by the same group that does NutraSweet, and it's called NutraSweet Neotame. It is primarily used in commercially prepared foods. So soft drinks, for example, or in other places where you are adding high-level sweeteners. And these sweeteners, some of them have aftertastes. Some of them don't do well under high heat, so you can't cook with them. Some of them have very unique flavor profiles. So often what happens is manufacturers blend sweeteners together so that each masks the challenges that are present with the others. But I don't think you can actually buy NutraSweet Neotame in the stores, but you are eating it. It is in your foods. Julie.
What's galactose and what's it found in?
Yeah, so galactose is another monosaccharide, and galactose and glucose form lactose. They form the milk sugar. Okay. So it is one of the two key components of the sugar that you find in milk.
What are the healthiest artificial sweeteners?
Oh, what are the healthiest artificial sweeteners? So we're going to talk a little bit about that in a few slides. And that is a bit of a challenging question to answer because it's going to depend on what kind of health benefits you are interested in. So if you'll hold me for right after the break, I'm going to touch base on that. Okay, one last question.
See, that is by weight. So if you took sugar by a level teaspoon and compared it to a level teaspoon of some of these others, it should be less.
That is a great point. That you've seen up here that I've got it sweetness by weight. That is not necessarily the same as volume. And some of these have a higher, they're fluffier. Some of these are more dense. So a tablespoon or a teaspoon of one and a teaspoon of the other are not equivalent amounts. In fact, you'll see on the next slide, when we start talking about how these compare to sugar, you're going to see that in some cases, like one packet is equal to multiple equivalent packets of sugar because of the sweetness difference. Okay, so we're going to cover these 4 sweeteners. I will circle back to you. Hey, Mark, I will circle back to you in just a second. No, actually, I'll go on ahead. What's your question? Yes, what is dextrose? If you will wait until we get to our 3rd sweetener, I will be able to answer that question. All right? So, first of all, let me say this. No one in this room is required to taste any or all of these. Let me be real clear. If you would rather not taste any of these, you are certainly welcome to just let them go. Number 2, I want you, if you choose to taste them, I want you to think about the profile. How does it first taste? What's the first sensation you register? As it dissolves on your tongue, how does that sensation change? Does it change? Some of you are going to experience these in very different ways. Not everyone is going to have the same sweetness and flavor profile. So in your bag, I'm going to have you, when I tell you to, I'm going to have you take everything out, and I'm going to have you set your water bottle on the floor. You're going to use this to take sips of water in between our sweeteners to cleanse your palate. And then you've got a plate that you're going to put all of your sweeteners on. So go ahead and open your bag and take everything out. In this instance, you can eat the science. That is the goal. Those of you that are playing along at home, whatever sweetener you might have in your pantry or in the school cafeteria, go and grab that, and then let's bring it back and compare. Okay, you've got a wet wipe, because I'm going to actually have you do this the old-fashioned way. You're going to dip your finger into these packets and taste them. And then, so I would suggest that you wipe your finger before we get started, and then you can choose to wipe your finger as we move our way through. And again, if you would rather not do this, that is totally fine. If you would rather just pour the entire packet in your mouth, that is also your prerogative. Okay, we're going to start with our Domino's pure sugar. This is cane sugar, so this comes from sugar cane. It is sucrose. And this is the way that I would recommend. So your packets run this way or this way. I'm going to recommend, for ease of getting your finger in, that you open them along the long length. And then once you've tasted it, you can put it in the trash bag so that we don't end up with sugar crystals all over everywhere. So going ahead and open, and then I'm going to have you taste. And again, I want the sensation of what's it feel like? What's the mouth feel? What's your first taste sensation? How does that change over time? What's your final sensation? The sweetness sits at the tip of your tongue. So if you stick it way back in the back, you're not going to taste much. Some of you are really tempted, just like with the butter, to just continue tasting the table sugar. This is probably familiar to most of us. Sucrose, there's 2 grams of sugar in this packet. And that's about 2/3 of a teaspoon of sugar. And in a teaspoonful, there are 16 calories. So just for comparison's sake. So 2 things I want you to pay, 3 things. This is sucrose, 2/3 of a teaspoon in this package, and 16 calories per teaspoon. Okay, now we're going to shift to honey. Now, those of you who have had honey before, you know that this is going to be sticky. In this case, you might just want to squeeze a little bit out onto your finger or directly into your mouth, whatever you want. This is, there are all sorts of brands of honey. My daughter for Christmas gave me a set of 4 different honeys from Brazil, each from bees that visit different flowers. And the differences among the honeys are unbelievable, absolutely amazing. This is just commercial grade honey. I can't tell you where it's from, I can't tell you what the bees visited, but I tore it at the top down. There you go. So that's got a very different flavor profile. It's got some strong overtones in it. And it lingers. It's thicker. Yeah, much more. It's a viscous. All right. I'm going to take a sip of water. Okay, so honey is 41% fructose and about 36% glucose. The type of honey gives you slightly different flavor profiles, slightly different ratios of glucose to fructose. It's really sticky. There are 2 teaspoons of honey in the packet that you've got in front of you, and honey has 22 calories per teaspoon. So a higher caloric load than an equivalent amount of table sugar, of Dixie crystals.
What about energy level at the same amount, same volume? How much more energy level are you getting between the 2?
The energy level that you're getting, so you're actually, so this is a measure of the, so it has more energy. There's more energy in that equal amount. Yeah. Okay, so now let's talk about Splenda. Now the Splenda packet, this is one I really would open along the long end because this short end is really narrow and you cannot get your finger in there. Splenda, remember, is sucralose. This is the molecule that looks a little bit like sucrose, but we've got the chloride, the 3 chloride molecules in its place. I want you to take a look at this. I want you to notice the way it feels, the thickness of it. Wait to see if you've got an aftertaste from it. It's not as grainy. It's much more much finer powder. For me, it has a little bit of a metallic aftertaste, but for some people, it has no aftertaste at all. So it's just the way that we each perceive the sweet. So Splenda is sucralose. Is sucralose. It has 1 gram per packet, which is 1/4 of a teaspoon. There is less in this packet than of your sugar packet. Significantly less volume in this packet. 1 packet, this 1/4 teaspoon, is equal to 2 teaspoons of sugar. So that means that if you put 2 packets of Splenda in your coffee, for example, you're putting the equivalent of 4 teaspoons of sugar into your coffee in terms of the sweetness level. It has 2 calories per teaspoon. Now you're going to notice it says up here, no calorie sweetener. How does it have 2 calories per teaspoon? This comes back to the question that Mark asked. Because sucralose is so many more times sweeter than sugar, you need a tiny, tiny amount of it. But to put like a few grains of powder into your drink, people are like, wait, there's nothing here. There's something wrong with this. So the manufacturers add a bulk filler to mimic the amount of sugar. And you can see it's still a tiny amount, but it has a bulk filler. It has something called dextrose, or it might have maltodextrin. These are sugars from corn. So they are corn starch that's been broken up into some individual sugars. It has a tiny, tiny sweetness factor. It's not terribly sweet. It doesn't have a lot of calories, but it still adds 2 calories per teaspoon. But as long as you're below 5 calories per teaspoon, you can go with no calorie or 0 calorie. So it isn't quite no calorie, but it falls below the threshold that makes it no calorie. So that's your bulking agents, dextrose, maltodextrin. A lot of artificial sweeteners and a lot of foods have dextrose or dextrin in it. It is to mimic the consistency and the mouthfeel of sugar without adding a lot of extra calories. Okay, now let's go with stevia. Last one. Ooh, I need some water. The dance of the millions is the phrase used to describe the boom and bust prosperity associated with the rapid of sugar prices in Cuba at the end of World War I. I guess they danced because they thought they had millions of dollars as sugar prices rose, and then they were sad when they lost all of their money after that. Okay, stevia in the raw. Again, a thin packet, not a lot in here. Open it long ways. So this is where some of you have no problem with this at all, and some of you feel like you just put something really awful in your mouth. Because some of us taste stevia in really visceral ways. It's like, whoa, that's like an assault on my mouth. And some of you don't taste it that way. Show of hands. How many of you were like, boom, wham? Okay. And some of you were like, nope, this was totally fine. I was all right with that. No problem at all. Yeah, so very different profiles for us. So stevia, again, is a natural product. It is a sweetener that is produced from the stevia plant. 1 gram per packet, again, 1/4 teaspoon. 1 packet is equal to 2 teaspoons of sugar. 2 calories per teaspoon because it uses dextrose again as a bulking agent. This was even more finely powdered than the Splenda. So very different profiles. Now again, when you dissolve these in your beverages, it tastes different yet again. Dissolving stevia in your tea or your coffee is very different than tasting it straight on. Some of these break down at high temperatures so you can't bake with them. Some of them you can bake with. So lots of different variety. So we've talked about monosaccharides, we've talked about disaccharides, 2 put together. And now we're going to spend a lot of time talking about polysaccharides. These are hundreds or even thousands of sugar molecules put together in these long polymer-like chains. They are essentially banks of carbohydrates for your body. For plants and animals, these polysaccharides, starch and glycogen, are the way that your body stores these sugars for long-term use. So enzymes in our bodies or in plant or animal cells, when needed, when our body needs the glucose, because glucose, again, is the major currency for your body. Your brain is a huge consumer of glucose. Those cells take the glucose in, break it down, get energy from it. So when your body needs glucose, it can go to these carbohydrate banks, starches and glycogen, and break some chains and get some glucose to be used. Potatoes and grains, wheat, corn, and rice, are the major source of starch in your diet. And there's a lot of stuff on this graph. I'm just going to touch on it really briefly. Starches, which are found in plants, there are 2 key kinds, amylose and amylopectin. They're both made of glucose. The difference is that amylose is just a straight chain, glucose, 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 glucose. It actually forms a helical structure that you see here. Amylopectin actually not only is glucose, 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 but it also branches off this way. So it forms straight and branched chains. And so your structure looks like this, where you can see every 10 or so glucose molecules, you get a branch. So these are the two things, the two ways that glucose is stored in starch, in your corn, in your wheat, in your rice, in your potatoes, as amylose and as amylopectin. Animals store their glucose not as starch, but as something called glycogen. It's the same glucose molecule. It has a whole lot more branching. And so you can see here it's got lots and lots and lots of branching. Here's part of the reason why. You and I may have an immediate need for a lot of glucose really quickly. Let's say that we suddenly are running from a bear. Yeah, hopefully I am not running in this, but all of the enzymes that can break down the starches, that can break down the glucose, sorry, not starches, all the enzymes that can break down these glucoses one at a time. Do you see all the different ends that they can start with and break it down? So I can very quickly break down lots of glucose molecules. Plants do not need to run from bears. So it's okay when they have something where I am breaking it down from this end, and I'm breaking it down from this end, and I'm breaking it down again from this end, and breaking it down again from this end. You and I have a need for glucose fast. And this branched structure gives us that quick access to the glucose that we need. So our bodies store glucose as glycogen. Plants store it as starch, amylose and amylopectin. And we store it in our liver and in our muscle cells. Your body has about a day's supply of glycogen. We make sure that I've actually got that right now that I've told you that. Yes, glycogen stores are depleted in about a day unless we replenish them by eating. Okay, let's talk about carbs, diet, and health. Bum, bum, bum. So when you eat carbs, when you eat grains, when you eat breads, when you eat potatoes, when you eat French fries, when you eat sweet pastries, your body breaks those complex carbs down into disaccharides and then further into monosaccharides. So this all takes place in your digestive system. They are absorbed in your small intestine. They travel to your liver where all of the sugars, glucose, fructose, galactose, any of those are converted all into glucose. So all these different sugars, fructose, for example, is converted into glucose so that all the carbs that you've eaten ultimately find their way as glucose, and then they travel through the bloodstream and they're absorbed by your cells. So that's the process of what happens when you eat carbs. Simple carbs, mono and disaccharides, these are the carbs that are absorbed really quickly. They give you a quick energy burst. This is what you find in candy and what you find in soft drinks and what you find in cookies. The cookies. You right now are breaking down and digesting a lot of simple carbs. And they're giving you quick energy, so you are absorbing everything we're talking about and you are going to drive your way home, but they are empty calories. As opposed to complex carbs, which are absorbed and broken down much more slowly, like whole grain, which are it is not a quick breakdown. They're polysaccharides. Your body's got to break those down. It takes time to do that. So they provide a sustained level of energy for you as opposed to that quick sugar rush that you get with your simple carbohydrates. Now, here's what happens in your body. The amount of glucose that's in your diet. If you don't get enough glucose in your diet, your body depletes your glycogen store, and it's got to find more glucose. So it's going to go to the proteins, or it's going to go to the fats that are in your body, and it's going to break those down. If you've got an appropriate amount of glucose in your diet, not too much, your body uses what it needs, and it can store up to 2,000 calories as glycogen, those excess stores. If you're getting more glucose than this in your diet, your body is storing that as fat. So lots and lots of carbs translates into lots of excess. The cup runneth over. You've stored as much as you can as glycogen, and now the rest of it you're storing as fat. That's the challenge. That's why people who often want to lose weight, where do they cut? What do they tend to cut right off the bat? Carbs. Sometimes, depending on the carbs you cut, that can be beneficial, especially if you're cutting these. If you're cutting out a lot of the simple carbs, the candy and the sodas, the empty calories that don't have any other benefit. I'm going to make some of you really angry, but long-term, zero-carb diets are not healthy. You can lose weight fast, but long-term, you've got to have some carbs providing the nutrients and the energy that you need. We have vilified carbs just like we've vilified fats. And as we talked about in our first week, you need some amount of fat in your diet in order to carry out the daily functions of your body. So again, we just want to be really careful about what and the amounts that we put in our bodies. There's all sorts of science or all sorts of myth and all sorts of pseudoscience and myth about what are good carbs and what are bad carbs and what are high glycemic and low glycemic and what are low carb versus low fat diets. And I am not going to dig deep into that. We just don't have time for that. But I will say this. A 2018 study in the British Medical Journal said this. Many people are confused about carbohydrates, but keep in mind that it's more important to eat carbs from healthy foods than to follow a strict diet limiting or counting the number of grains of carbohydrates consumed. So what are you putting in, and are you being generally mindful of the amount you are putting in versus being so incredibly strict for incredibly long periods of time? All things in moderation, including Jackson Center cookies. All things in moderation. So let's talk about the health issues of sugar substitutes. I told you we'd touch base on this a little bit. Here are some of the potential health issues. And again, I say potential health issues. These have all been approved by the FDA. They've all been subject to rigorous testing. But when we eat them for decades, and we eat them at very high levels, when you are drinking 6 and 7 Diet Cokes a day, there are some unknown effects at that kind of concentration. Here's one that seems to be pretty backed up by a lot of the science. When you take in a lot of artificial sweeteners, you are changing the makeup of your gut microbiome because you are now providing them with an energy source that is different than what they are used to seeing. Your body cannot break down these artificial sweeteners, so they pass into your gut, but many types of bacteria can. And if you're consuming lots of these artificial sweeteners, you are providing a bonanza for those bacteria that can break this down. But remember, for most of our human life, most of our time here on Earth, these molecules have not been part of our regular diet. They can interfere with the responses about how we deal with glucose control and energy regulation because they are telling your tongue is telling your brain sweet is coming and no calories are coming from that. And especially if you're doing a lot of these ultra high sweeteners, you are essentially continually signaling to your brain, there's a whole lot of sweet coming and there is no sweet coming. So your brain dissociates, your body dissociates the I am eating sweet from the how do I actually regulate energy? How do I prepare for what is coming from that? It can throw off the typical rhythm. Now let's talk about cancer. There was a lot of data when saccharin first came out, when sweet and low first came out, that it caused cancer in lab rats, that it caused bladder cancer in lab rats. And that data is absolutely true. Saccharin causes bladder cancer in lab rats. Saccharin does not cause bladder cancer in humans because lab rats or not, have a different substance in their urine that humans don't and a different pH, and only in the rat did saccharin with that specific substance in the urine actually lead to a carcinogen. So yes, saccharin causes cancer in lab rats, but it causes cancer by a mechanism that does not exist in humans. So I'm not saying that saccharin is what you should all run out and have, But we have the most accumulated data because saccharin is the sweetener, the artificial sweetener that's been around the longest. And the data that says it causes cancer has been pulled off of all of the FDA regulation. So the other artificial sweeteners, I don't have data that says they do or don't. In the FDA studies, they did not. But again, you've heard me talk about, we've talked about cancer before. Cancer can take decades to form as you have this accumulation of genetic hits. I am not saying artificial sweeteners cause cancer. I am saying we need longer windows of time to make some assessments about that. They are not always effective against obesity and diabetes. So when people take artificial sweeteners to diet, often they are not effective. And one of the reasons that they're not effective is because we think, oh, I am having a diet beverage, I can have an extra Oreo. This is the same thing we talked about in week 1. I am having a low-fat cookie, I can have 5 of them. What is it that in order to replace the taste of fat, we increased in many of our low-fat foods? Sugars. And not complex carbs, simple carbs. So again, it's this mindset. Our brain, we allow ourselves to take these kinds of shortcuts. And as of 2018, there is no clear data that says that non-sugar sweeteners are unsafe. But there's also no clear data that says they lead to improved health outcomes. Now, that's not to say that some of you in this room that have given up sugar have not had improved health outcomes. That's not what I'm saying. And some of you, because you may have diabetes or you have something else, you've had to step away from sugar and you use one of these artificial sweeteners. Maybe it's stevia, maybe it's monk fruit. We didn't even talk about monk fruit. But I'm saying that in broad scale, we have no clear data that the use of artificial sweeteners reduces obesity in this population, in our population. That's a sobering thought because that's what we've been sold. That I use diet beverages to help me lose weight. But we don't have the long-term data that says that that makes a real difference. Okay, I'm going to shift gears. We've talked a lot about sugar. We've talked a lot about artificial sweeteners. So these are the storages that we talked about, our starches and our glycogen. And you can see here they're all glucose and they're all called alpha glucose. This is the structure of alpha glucose. The molecule behind me is alpha glucose. I want you to pay attention to on the first carbon, the OH group sits below the ring. Here it is. On the first carbon, the OH group sits below the ring. That's the position of alpha. This is what is called alpha carbon. This is beta carbon. Do you see the difference? The OH group doesn't sit below the ring. It sits above the ring. So it would sit here. That difference, that single position difference, is the difference between the sugars that we can consume, the glucose that you and I can break down, and the glucose that we can't break down that's in cellulose, that forms fibers like cotton fibers. So, a completely different polysaccharide, many, many repeating units of glucose, is cellulose. It is for all the world identical to the sugar that you and I have just tasted, except this OH sits above the ring, and you and I don't have the enzymes that break this structure apart. So in our digestive system, we don't have an enzyme that breaks two glucose molecules that are joined together when the OH sits above the ring. That's why we don't get any nutrients when you eat cellulose, like celery fiber. Because your body can't digest it. There's a whole lot of glucose in there, but your body can't break it down. Now, the bacteria in your gut can. The bacteria that live inside a termite can break down cellulose in the wood. Cows go through a multi-chewing process where they're breaking it down and there's bacteria in one part of their stomach. Rabbits do this because the enzymes that break down cellulose in their system are in their large intestine. So it goes through their small intestine, into their large intestine. They break down the cellulose. The rabbit poops, and then the rabbit turns back around and eats it, and it's already broken down the cellulose, so as it digests the feces, it now gets energy from it. Not something I would recommend that we try. But different organisms have adapted to different ways to extract energy. Some of you are now just totally disgusted about this whole thing. This is why we did the taste test before the break. But that one difference, we're talking about patterns of chemicals here. This one difference is why you and I can't break down celery, and we can't break down cotton fibers, and we can't break down grass, because we don't have the enzymes to break that apart. So, cellulose is the most abundant polysaccharide, the most abundant long chain of sugars, and the most abundant biomass on the earth. There are tons of cellulose on our planet. It's an important structural component of the cell wall of green plants. So it provides much of the structure, much of the rigidity of the cell wall around plants and of stems and of tree trunks. That cellulose is woven in with other things like lignin and hemicellulose, and it provides the structural stability that we see in plants. It's kind of like the skeleton. There's a really similar version of cellulose called chitin. Chitin is found in shells of like shellfish. It also gets broken down, and we use it as N-glucosamine, which we use for our joints. But it's also a polysaccharide, not of glucose, but of a similar molecule called chitin. So here's your cell wall, and here is you've got, in gray, you've got lignin, in green, you've got hemicellulose. Hemicellulose is a lot like cellulose. It's a slightly different structure. It's a shorter structure. And then here is your cellulose. And you can see these are glucose units, just repeating many, many, many glucose units. Up to 10,000 glucose units in a single fiber of cotton. Wood pulp, anywhere between 300 and 1,700 glucose repeating units in that chain. So just hundreds and hundreds of glucose units, but it's beta glucose, not alpha glucose, so you and I can't extract energy from it. And then these individual cellulose chains are joined together by your friend and mine, hydrogen bonds. We talked about hydrogen bonds last week. We talked about hydrogen bonds the week before. We'll talk about hydrogen bonds next week. They are what hold all of these cellulose molecules together. They're what make up the multiple fibrils in a cotton fiber. Multiple fibrils of cellulose held together by hydrogen bonds, all pointing them in the same or in a similar direction. So the cellulose content of the fluffy fiber of the cotton, 90% of that cotton fiber is cellulose. So when you see the cotton bolls out in the fields, you've got the seeds, but then all that fluff, that is almost all cellulose. It is almost all repeating units of glucose. It's just glucose in a form that you and I can't chew up and get any energy from. Okay. Let me close us with this, because I spent a lot of time standing up here dressed in 1700s or early 1800s English costume. And we've had a lot of fun, and we've tasted different kinds of sugars, and we've talked about the value of cotton fibers and cotton fabric. But I'd be doing us a huge disservice if I didn't talk about the fact that in the 1700s and the 1800s, the rise of sugar consumption and the use of cotton as a textile for the Industrial Revolution was brought about by the enslavement of millions of people from Africa. And I want to make sure that we spend at least a little bit of time acknowledging that, because that is another way that these molecules impact human history. So there were 10 to 12 million enslaved Africans in the period from the 1500s to 1808, when the slave trade, the transatlantic slave trade, was declared illegal, was disallowed. This map shows you most of the places where those Africans were taken from and where they were taken to. And what you can see is that the vast majority of them were taken to the Caribbean and to Brazil. And they were primarily brought into slavery to work the sugar plantations. Probably 2/3 or more of those 10 to 12 million African slaves were taken to sugar plantations. Sugar extraction was a back-breaking, hot job. And when the Europeans landed on these islands and started planting sugarcane, they took the native population and forced them into service. The native population also quickly succumbed to all of the diseases that the Europeans brought with them, and there weren't enough workers to do the job needed for the sugarcane, so they turned to Africa and they brought over Africans enslaved to work these sugar plantations. This is a group of Africans, a group of slaves working a sugar plantation owned by the Dutch. They're crushing the sugarcane, they're extracting the juice, then there are multiple rounds of it being boiled. And when it's boiled, then you've got to skim off the impurities, and then you've got to boil it again, multiple rounds, and then you've got to pack it into those sugar cases, and you've got to send them off to Europe. Of the 10 to 12 million slaves that were taken from Africa, not even 400,000 of them were brought to America. That was a completely stunning figure for me, that the vast, vast majority of them were brought into the West Indies to work originally the sugarcane, and then the tobacco, and in some cases the rubber plantations. That 388,000 Native African Americans that were brought as slaves to America really initially peaked in the mid-1700s, and then those numbers started to fall at the end of the 1700s. And then there was a really sharp uptake in like the last 15 years before the slave trade was outlawed. And that was because of what I'm about to show you next. This was cotton production in our country in millions of pounds. 1.5 million pounds in 1790. By 1860, 2,275 million pounds of cotton were produced in this country. And the vast majority of that cotton actually went to the textile mills of Europe. 77% of the cotton consumed in the British textile mills came from the United States, came from the South. 90% of the cotton from France, 60% in the German region, and 92% in Russia. So there was an enormous appetite for raw cotton in Europe that was fulfilled by the U.S. South. I told you in about 1820, sugar no longer was the #1 import into Britain. Raw cotton was, primarily from the United States. And I also just told you 388,000-ish. Now those numbers are based on the a slave trade organization that accumulated that data. That does not include the individuals that did not survive the passage from Africa to the United States, called the Middle Passage. 388,000 brought here. But then, at the end of the 1700s and into the 1800s, we shifted from bringing slaves over from transatlantic slave trade to domestic slave trade. So the concept of selling a family downriver was where the Deep South got most of the slaves that they needed to run the cotton plantations. So more than 1 million slaves were traded from the North and from the Upper South into the Deep South to keep the cotton plantations moving forward. And even though the northern states had outlawed slavery, The textile mills in the northern part of the United States were dependent upon the cotton that came from slave-produced southern states. So there was this unholy alliance between the economy and the Industrial Revolution that kept it very, very profitable to be a slave owner, especially if you were on a cotton plantation. The cotton gin invented by Eli Whitney made it easier to pull the seeds out of the cotton, and the cotton gin could do the work of 50 people. So now you could grow more cotton, and you could more quickly get your raw cotton and send it off to England. So again, I just want to acknowledge, as we spend time talking about sugar and about the sugar trade, and as we spend time talking about cotton and how it drove the Industrial Revolution, that there was an enormous cost that was borne in making this a reality. And I would have been foolish to gloss over that and not acknowledge that if we're talking about the impact that these molecules have on history. Key take-home points as we wrap up. Carbohydrates are polymers of sugar. They are repeating units of sugar. They're used by your cells for fuel, and they're also used, especially by plants, to provide that structural material, that building support like cellulose. We talked about monosaccharides. That's a single unit of sugar. We talked about glucose, fructose, and galactose. We talked about disaccharides, which are two sugars that come together. Two glucose molecules is maltose. A glucose and a galactose is lactose, which you find in milk. A glucose and a fructose is table sugar. That's sucrose. And then we talked about when you have a whole bunch of sugars, polysaccharides. In plants, those polysaccharides are stored as starch. In animals, they're stored as glycogen. And then we also talked about cellulose, which is used by plants, but that you and I can't access the glucose molecules in that. The other thing that I want us to take home, we talk a lot about patterns and about differences without digging deep into the chemistry. The arrangement of these atoms provides the reason why some things are sweeter than others, why some things are used by energy and in animals and others can't be, and all the different properties. It all comes down to chemistry. What atoms are in what arrangement and in what specific order. That is the beauty of chemistry. You shift one molecule, you shift one atom around, and the molecule completely changes, now has a completely different function. Moving this up here completely changes us, takes us from something that you and I can consume to cotton. And we talked about sugar substitutes, and those sugar substitutes can be man-made, they can be artificial, created in labs, like sucralose or like saccharin, or they can be natural. So they are naturally found, and we've incorporated them for our use. They have varying characteristics. They have varying flavor profiles, as you learned tonight. They have varying uses, and they have a challenging place in our relationship with carbohydrates. So again, I just want us to be aware that anytime we go to extremes in any part of our diet, we run into challenges. If I cut out all fats, if I cut out all carbs, we run into challenges there. Okay, next week, we're going to wrap up Biotech 201. We're going to talk about nucleic acids. And we're going to talk about these four individuals. And this is going to be a lesson in workplace dynamics gone wrong. How not to behave with your colleagues. So it's really interesting. We've got a really fantastic activity that the group will do. Those of you that are watching at home, you've got an email that tells you how you can get some of the raw materials that we're going to need for next week's activity. You can come to Hudson Alpha and you can pick up your kit, and then there's some things that you've got to buy yourself to add in. But I hope you get the chance to do that. We are going to have a whole lot of fun as we tie all of these pieces together. So with that, Be safe going home. I think it's probably started raining again. Thank you for being with us tonight. Have a good night, everybody.